5 times in the history of this planet, almost everything alive has died, not gradually, not in the background, catastrophically, in geological blinks so sudden and so total that the fossil record reads like someone pulled the plug on an entire biosphere and walked away. Ice ages that froze the oceans from pole to equator, volcanic eruptions that poisoned the atmosphere with sulfur for millions of years, an asteroid that turned the sky into an oven and shut out the sun for a decade. Each of these 5 mass extinctions wiped out between 75 and 96% of all species on Earth. Each one reset the biological clock and forced life to start over from whatever was left, and each time, almost nothing survived. The question is not why so many things died. Death at those scales is the default. The question is why anything survived at all. What does an animal need? What specific combination of traits, of biology, of sheer dumb luck, to walk through the fire 5 consecutive times and come out the other side still breathing? Because there is an animal that did exactly that. It is alive right now. You can find it on the beaches of Delaware and the coastal mudflats of Southeast Asia. It looks like a military helmet with a tail. It is older than trees, older than flowers, older than every dinosaur that has ever lived and every mammal that has ever drawn breath. It has survived every single mass extinction event in the history of complex life on Earth, all 5 of them, back to back, across 450 million unbroken years. Its name is misleading. Its anatomy is alien. Its blood is blue, and for the last 60 years, it has quietly been saving your life without you knowing it. This is the horseshoe crab, and the story of how it survived everything the planet has thrown at it, and what is threatening it now, is one of the most extraordinary survival stories in the history of biology. The first thing to understand about the horseshoe crab is that it is not a crab. It is not a crustacean at all. Despite its common name, the horseshoe crab belongs to the subphylum Chelicerata, the same group that includes spiders, scorpions, ticks, and mites. Genetic analysis published in Systematic Biology confirmed what comparative anatomists had suspected for over a century. Horseshoe crabs are not just related to arachnids. They are nested within the arachnid family tree. They are, in the most literal phylogenetic sense, aquatic spiders, or at least the closest living aquatic relatives of the lineage that produced spiders. They have chelicerae instead of jaws. They have book gills instead of the internal gills of true crabs, structures that are anatomically homologous to the book lungs of spiders and scorpions. They have no antennae, and they have 10 eyes, not 2, not 8, but 10 distinct light-sensing organs distributed across their body, including a pair of lateral compound eyes, a pair of median simple eyes, and additional photoreceptors on the tail and along the ventral surface. The horseshoe crab does not just see light, it senses it from almost every direction simultaneously. There are only 4 living species. Limulus polyphemus, the Atlantic horseshoe crab, ranges from Maine to the Yucatan, along the eastern coast of North America and the Gulf of Mexico. 3 additional species, Tachypleus tridentatus, Tachypleus gigas, and Carcinoscorpius rotundicauda, inhabit the coastal waters of Southeast Asia, from the shores of Japan and Korea through the South China Sea to India. 4 species. That is the entire global census of an animal lineage that has been continuously present on this planet since the Ordovician period, an era so ancient that when the first horseshoe crabs appeared, the most advanced life on land was moss. Trees did not exist, flowers did not exist. The first insects were still 100 million years in the future. The first vertebrate would not crawl onto land for another 50 million years. And yet the horseshoe crab body plan that appears in the fossil record from those earliest times is recognizable as the same animal you can watch spawning on the beaches of Delaware Bay every May. Jurassic-era fossils from 148 million years ago, found in a quarry in Solnhofen, Germany, and in Poland, are virtually identical in shape, proportion, and size to modern Limulus polyphemus. They have the same horseshoe-shaped prosoma, the same hinged opisthosoma, the same long telson tail spine, the same arrangement of book gills on the underside. If you placed a modern Atlantic horseshoe crab beside one of these Jurassic specimens, you would struggle to tell them apart. an animal that lived alongside Archaeopteryx and Stegosaurus, in an ocean where ichthyosaurs and plesiosaurs were the apex predators, looks essentially the same as an animal you can pick up on a New Jersey beach today. That morphological conservatism, the fact that the basic body plan has barely changed across an almost incomprehensible span of geological time, is not evidence of evolutionary stagnation. It is not a failure to adapt. It is evidence of a design so effective, so perfectly matched to the demands of its ecological niche, that evolution has found almost nothing to improve. The horseshoe crab body plan works. It has worked since before the first forest grew on land, since before the first vertebrate crawled out of the water, since before the first insect took flight, since before the first dinosaur hatched from an egg, and it has kept working through 5 successive planetary catastrophes that destroyed everything from the trilobites to the non-avian dinosaurs, animals far more famous, far more powerful, and far more specialized than a slow-moving, bottom-feeding chelicerate eating worms in the mud. To understand how remarkable that is, you need to walk through what the horseshoe crab actually survived, not as an abstraction, because 5 mass extinctions is easy to say and impossible to feel, but event by event, catastrophe by catastrophe, extinction by extinction, only then can you appreciate the sheer range and severity of what this animal endured while everything around it died. And the horseshoe crab was not entirely alone in this achievement. A handful of other lineages share parts of its survival record. The nautilus, a cephalopod with a coiled shell, now confined to the deep waters of the Indo-Pacific, traces its ancestry back roughly 500 million years, though the modern genus is far younger than the lineage. Lingula, a small burrowing brachiopod found in the mudflats of Asia and Oceania, has survived virtually unchanged since the Cambrian and endured every mass extinction event in the history of complex life. Sharks, as a group, have persisted for over 450 million years, though individual species and families have turned over dramatically at each extinction boundary. But among animals large enough to see with the naked eye, few can match the horseshoe crab's combination of lineage age, morphological consistency, and unbroken survival through all 5 of the planet's great biological catastrophes. The first was the late Ordovician extinction, roughly 444 million years ago. The horseshoe crab lineage was already ancient by this point. It had appeared in the fossil record at least 6 million years before this event. The Ordovician extinction was caused by rapid glaciation. The supercontinent Gondwana drifted over the South Pole, triggering a massive ice age that locked up enormous volumes of seawater in continental glaciers. Sea levels dropped by over 100 meters in some estimates, a catastrophic withdrawal that drained the shallow continental shelf habitats where the overwhelming majority of marine life existed. The warm, shallow seas that had supported the great Ordovician biodiversity radiation, the most explosive diversification of marine life up to that point, simply disappeared. Trilobites were decimated. Brachiopod diversity collapsed. Graptolites, bryozoans, and corals were devastated. The reef ecosystems that had spread across the shallow shelves were destroyed. 86% of all species vanished in 2 distinct pulses, one from the glaciation itself, and a second from the rapid warming and ocean anoxia that followed when the ice retreated. The horseshoe crab survived both. The second was the late Devonian extinction, roughly 372 to 359 million years ago, the extinction that killed Dunkleosteus and every other placoderm on Earth. This was not a single event, but a prolonged crisis spanning millions of years. The great Devonian reef systems, which had been the most complex marine ecosystems the planet had ever produced, collapsed entirely. Ocean anoxia, vast dead zones of oxygen-depleted water, spread across the continental shelves, suffocating everything in their path. The cause is still debated. Accelerated chemical weathering from the first forests on land drawing down atmospheric KNEC and flushing nutrients into the seas, triggering algal blooms that consumed the oxygen in the water column. 75% of all species went extinct across 2 major pulses, the Kellwasser and Hangenberg events. The armored fish, which had dominated the Devonian oceans for 60 million years, were completely eliminated. The horseshoe crab survived. The third was the Permian Triassic extinction, the Great Dying, roughly 252 million years ago. This was the worst biological catastrophe in the history of life on Earth, and it is difficult to overstate how close the planet came to losing everything. The Siberian Traps, one of the largest volcanic provinces ever documented, erupted for roughly a million years, flooding the atmosphere with carbon dioxide, sulfur dioxide, methane, and toxic heavy metals. Global temperatures rose by as much as 10 degrees Celsius. The oceans warmed, lost their oxygen, and acidified. Hydrogen sulfide, a toxic gas produced by anaerobic bacteria in oxygen-depleted water, may have bubbled out of the dying oceans and into the atmosphere, destroying the ozone layer and poisoning the air itself. 96% of all marine species went extinct. 70% of all terrestrial vertebrate species vanished. Reef ecosystems collapsed so completely that it took 10 million years for reef-building organisms to recover. The fossil record in the earliest Triassic shows a world that was, by every biological measure, nearly dead. The horseshoe crab survived, and it was not alone. Lingula, a brachiopod genus that has persisted virtually unchanged since the Cambrian, not only survived the Great Dying but thrived in the devastated aftermath, becoming one of the most abundant organisms in early Triassic shallow marine environments. The same pattern, repeated. The survivors are not the strongest. They are the most tolerant. The fourth was the Triassic-Jurassic extinction roughly 201 million years ago. Massive volcanism from the Central Atlantic magmatic province, associated with the rifting of the supercontinent Pangaea, triggered rapid climate change, ocean acidification, and widespread habitat destruction across both land and sea. Roughly 80% of all species went extinct. The ecological vacuum left behind was filled by the dinosaurs, which would dominate terrestrial ecosystems for the next 135 million years. The horseshoe crab survived and continued essentially unchanged, while an entirely new dynasty of giant reptiles rose, diversified, and took over the planet. The fifth was the Cretaceous-Paleogene extinction, 66 million years ago, the one everyone knows. The Chicxulub asteroid, 12 kilometers across, struck the Yucatan Peninsula at 44,000 miles per hour. The thermal pulse broiled exposed surfaces. Impact winter shut down photosynthesis for nearly 2 years. Acid rain poisoned freshwater systems and the upper ocean. The food chain collapsed from the bottom up. 75% of all species were eliminated, including every non-avian dinosaur, every pterosaur, every marine reptile, and every ammonite. The horseshoe crab, ploughing through the sediment of the continental shelf, eating detritus and tolerating conditions that killed virtually everything else around it, survived. 5 extinction events, 450 million years, glaciation, ocean anoxia, supervolcanic eruptions, atmospheric poisoning, an asteroid impact that turned the sky into a furnace and shut out the sun for years. The horseshoe crab walked through every single one of them, not because it was powerful, not because it was fast, or intelligent, or aggressive, or capable of fighting back against any of the forces arrayed against it. It is none of those things. It is slow. It is simple. It eats mud. And that, paradoxically, is why it survived, because the horseshoe crab possesses a combination of biological traits so perfectly calibrated for endurance that they amount to an evolutionary survival manual, a formula that, once you extract it from the horseshoe crab's biology, explains not just why this one species survived, but why the vast majority of everything else did not. The formula has 5 components, and every one of them cuts against what we intuitively assume survival requires. We assume that survival means being the biggest, the fastest, the most powerful. The horseshoe crab demonstrates across 450 million years of evidence that survival means being the most tolerant, the most flexible, and the least dependent on any one thing going right. The first trait is habitat flexibility. Horseshoe crabs can live in shallow coastal waters, estuarine mudflats, brackish bays, and deeper continental shelf environments down to depths of roughly 60 meters. They tolerate a wide range of salinities, from nearly fresh water to full marine conditions, and a wide range of temperatures, from the cold waters of the northern Atlantic to the warm tropical shallows of the South China Sea. This matters enormously during mass extinctions because different extinction mechanisms target different habitat types. The Ordovician Ice Age destroyed shallow shelf habitats by draining them entirely. Any species confined to shallow water was stranded and killed. The Devonian anoxia killed organisms dependent on well-oxygenated water. Anything that needed oxygen-rich conditions to breathe simply suffocated. The Cretaceous asteroid devastated surface-dwelling organisms and those dependent on photosynthetic food chains. Anything tied to sunlight for its food supply starved in the dark. An animal that can survive in multiple habitat types, shallow and deep, warm and cool, salt and brackish, oxygenated and partially anoxic, has fallback options when any one habitat type is destroyed. Specialists die because their one habitat disappears and they have nowhere else to go. Generalists survive because they always have alternatives. The horseshoe crab is the ultimate habitat generalist. The second trait is dietary generalism. Horseshoe crabs are opportunistic bottom feeders. They eat marine worms, small mollusks, algae, detritus, crustaceans, dead fish, and essentially anything organic they encounter on or in the sediment. They do not depend on any single food source. They do not target any specific prey. They eat what is there. When mass extinctions collapse specific food webs, wiping out the plankton that feed the fish that feed the larger predators, a bottom-feeding detritivore that can eat whatever organic matter accumulates on the seafloor is insulated from the cascading collapse. And there is a darker truth here. During a mass extinction, when enormous numbers of organisms are dying simultaneously, the one thing that is not in short supply is dead organic matter. Detritus, decaying organisms, biological debris, the settling remains of a dying ecosystem, is the last food source to disappear in any extinction event. It is, in the most literal sense, the food of death. And for an animal that eats it, a mass extinction is not a famine, it is a surplus. The third trait is reproductive volume. A single female Atlantic horseshoe crab lays roughly 60,000 to 120,000 eggs per spawning season, deposited in multiple clutches in shallow nests dug into sandy beaches at high tide. The eggs are small, roughly 2 millimeters in diameter, and buried in sand, which insulates them from many of the surface-level environmental catastrophes that kill exposed organisms. Thermal pulses, acid rain, atmospheric toxins, ultraviolet radiation from a damaged ozone layer. A layer of sand provides meaningful protection against all of these. High-volume reproduction is a classic extinction survival strategy, and the mathematics are simple. Even if environmental mortality spikes to 99%, a species that produces 100,000 offspring per female needs only a tiny surviving fraction to repopulate. You do not need to be tough enough to survive a mass extinction individually. You need to produce enough offspring that some of them, somewhere, by chance or by shelter, make it through. The horseshoe crab plays this numbers game with overwhelming force. The fourth trait is metabolic tolerance. Horseshoe crabs can survive extended periods of low oxygen. Studies have shown they can tolerate hypoxic and even anoxic conditions that would kill most marine animals within minutes. They can survive wide temperature fluctuations from near freezing to warm tropical water. Their metabolic demands are modest compared to more active marine animals. They are slow, energy efficient, and do not require the enormous caloric intake that drives active predators to extinction when food supplies collapse. During the mass extinction events that killed through ocean anoxia, particularly the Devonian and the Permian, this tolerance would have been the margin between survival and suffocation. While faster, more metabolically demanding organisms died gasping in oxygen-depleted water, the horseshoe crab endured. The fifth trait may be the most remarkable of all, and it is the one that connects the horseshoe crab's ancient past to the most advanced technology of modern medicine: the immune system. Horseshoe crabs have an open circulatory system. Their blood-like fluid, called hemolymph, flows freely through open body cavities rather than being confined to a closed network of blood vessels, the way mammalian blood is. In most animals, an open circulatory system is a vulnerability. Any wound, any crack in the exoskeleton, exposes the entire internal fluid to the surrounding environment and every pathogen in it. For an animal that lives on the seafloor, burrowing through sediment that contains billions of bacteria per cubic centimeter, this should be a death sentence. One cut, one puncture, and the entire body is flooded with infection. Instead, the horseshoe crab evolved one of the most effective and most ancient innate immune responses in the animal kingdom. Its hemolymph contains specialized cells called amoebocytes, large, granulated cells that function as both immune sentinels and emergency response units. When an amoebocyte detects the presence of endotoxins, lipopolysaccharide molecules found in the outer cell walls of gram-negative bacteria, it undergoes a rapid degranulation response, releasing a cascade of clotting proteins that physically seal the infection site. Within minutes, the proteins form a dense, insoluble gel that physically traps the bacteria, immobilizing them and preventing them from spreading through the open circulatory system. The reaction is extremely fast, extremely sensitive. It can detect endotoxin concentrations measured in parts per trillion, and it works against an extraordinarily broad range of bacterial pathogens. It is not an adaptive immune response like the antibody-based system that mammals use. It is an innate system, hardwired, ancient, and indiscriminate. It does not learn. It does not need to. It simply detects and seals, with a speed and reliability that has been refined by 450 million years of natural selection in the most bacteria-rich environment on the planet. In a world where bacterial infection from contaminated sediment was a constant daily threat, and during mass extinction events when the ocean floor would have been saturated with the decaying remains of billions of dead organisms and the bacterial populations feeding on them would have exploded, this immune system was not merely useful. It was a survival advantage of the highest possible order. While other organisms died of secondary infections triggered by wounds, environmental stress, or compromised immune function, the horseshoe crab's amoebocyte system kept working, sealing every breach, trapping every invader, maintaining the integrity of an open circulatory system in conditions that should have made open circulation fatal. And that immune system is the reason the horseshoe crab matters to you personally, right now, whether you have ever seen one or not. In the 1960s, a pathobiologist named Frederick Bang was studying the circulatory system of horseshoe crabs at the Marine Biological Laboratory in Woods Hole, Massachusetts, when he made an accidental discovery that would change modern medicine. He noticed that horseshoe crab blood clotted dramatically, almost violently, in the presence of gram-negative bacteria. Not just any clotting. The blood formed a dense, insoluble gel that physically encapsulated the bacteria within minutes. Bang enlisted the help of hematologist Jack Levin, who identified the trigger. The blood was reacting not to the bacteria themselves, but to endotoxins, lipopolysaccharide molecules found in the outer cell walls of gram-negative bacteria. Even vanishingly small concentrations of endotoxin triggered the clotting cascade. Levin realized this reaction could be weaponized as a detection tool, a biological sensor so sensitive it could identify bacterial contamination at concentrations of parts per trillion. The result was the Limulus amoebocyte lysate test, LAL, named for the amoebocyte cells in the horseshoe crab blood that drive the clotting response. The test transformed pharmaceutical safety. Before LAL, the standard method for detecting bacterial contamination in injectable drugs was the rabbit pyrogen test, injecting the drug into rabbits and monitoring them for fever. It was slow, expensive, unreliable, and required large numbers of animals. LAL was faster, cheaper, more sensitive, and more specific. By the 1970s, it had been adopted by the US Food and Drug Administration as the standard endotoxin detection method for injectable drugs, vaccines, intravenous fluids, and implantable medical devices. Every injectable drug that has been approved in the United States in the last 50 years has been tested for contamination using a substance derived from the blue, copper-based blood of an animal that has been alive since the Ordovician period. Every vaccine, every IV bag, every surgical implant, all of it screened by the immune system of a creature that looks like a Paleozoic military vehicle. The blood is blue because it uses hemocyanin, a copper-based oxygen transport molecule, instead of the iron-based hemoglobin that colors mammalian blood red. When oxygenated, the copper ions give the blood a distinctive bright blue color. Hemocyanin is less efficient than hemoglobin at carrying oxygen in warm, well-oxygenated environments, but it functions across a wider range of temperatures, pH levels, and oxygen concentrations. Yet another trait that contributes to the horseshoe crab's extraordinary environmental tolerance and its capacity to survive conditions that kill more physiologically constrained organisms. The harvesting process is clinical and systematic. Each year, approximately 500,000 Atlantic horseshoe crabs are captured from coastal waters during the spawning season, transported to biomedical laboratories, strapped to racks, and bled through a needle inserted into the pericardial membrane, a soft spot on the underside of the body, near the heart. Roughly 30% of their blood volume is drained over a period of 24 to 72 hours. The crabs are then returned to the ocean. The industry reports that most survive. Independent research tells a different story. Post-bleeding mortality rates are estimated at 15 to 30%, meaning that of the 500,000 crabs bled annually, between 75,000 and 150,000 die as a direct result of the process, and the survivors do not come back unscathed. Studies have documented disorientation, reduced spawning activity, altered movement patterns, and decreased reproductive success in crabs that survive bleeding. The crabs that are returned are not the same crabs that were taken. This is where the story of the ultimate survivor becomes the story of the ultimate irony, an irony so precise it reads like a parable. An animal that survived 5 mass extinctions, that endured ice ages that froze the oceans, volcanic eruptions that poisoned the atmosphere for 1,000,000 years, ocean anoxia that suffocated everything in the water column, an asteroid impact that killed the dinosaurs, and 450 million continuous years of predators, competitors, diseases, and environmental catastrophes that eliminated 99% of every species that has ever lived, is now in decline. Not because of volcanism, not because of glaciation, not because of an asteroid, because of a single species that has existed for roughly 300,000 years. A species that, in the full context of the horseshoe crab's tenure on this planet, arrived approximately 30 seconds ago. The threats are multiple, they are compounding, and they are accelerating. Biomedical harvesting removes hundreds of thousands of breeding adults from the population every year during the spawning season, the worst possible time to remove reproductive animals from a population. Habitat destruction along the Atlantic coast, including coastal development, beach armoring with rock and concrete, pollution and dredging, is degrading and destroying the sandy spawning beaches that horseshoe crabs depend on for reproduction. Overharvesting for use as bait in the American eel and channeled whelk fishing industries removes additional animals from the population entirely. These crabs are not returned. And climate change is altering the temperature, salinity, and chemistry of the coastal waters where horseshoe crabs live, with uncertain but potentially significant effects on spawning timing, larval development, juvenile survival, and habitat suitability. No single threat is necessarily fatal on its own. Together, they constitute exactly the kind of slow, compounding environmental squeeze that has killed larger, more powerful animals than the horseshoe crab, including, as this channel has documented, the most dominant predators the planet has ever produced. The ecological consequences of horseshoe crab decline cascade far beyond the species itself, because the horseshoe crab is not just a survivor, it is a keystone species. Its eggs are a critical food source for migratory shorebirds, particularly the red knot, a small sandpiper that undertakes one of the longest migrations in the bird world. From the southern tip of South America to breeding grounds in the Canadian Arctic, a round trip of roughly 30,000 kilometers every year. The red knots time their northward migration to coincide precisely with horseshoe crab spawning on the beaches of Delaware Bay, arriving in enormous flocks to gorge on the protein-rich eggs and refuel for the final, grueling leg of their journey to the Arctic. Without that refueling stop, the birds arrive at their breeding grounds too depleted to reproduce successfully. Since the 1980s, as horseshoe crab populations have declined under the combined pressure of biomedical harvesting, bait harvesting, and habitat loss, red knot populations have collapsed by approximately 75%, a decline that tracks the horseshoe crab population curve with chilling precision. The red knot was listed as a threatened species in the United States in 2014. Its decline is not a coincidence. It is a direct ecological consequence of removing a keystone species from a system that evolved around it. There is a synthetic alternative to bleeding horseshoe crabs, and it works. Recombinant factor C, RFC, is a lab-produced protein that detects bacterial endotoxins with the same sensitivity as LAL, without requiring a single horseshoe crab to be captured or harmed. It has been commercially available since 2003. The European Pharmacopoeia endorsed its use in 2019. Eli Lilly, one of the world's largest pharmaceutical companies, switched to RFC in 2016 and reported that it was not only effective, but in some ways superior to LAL, fewer false positives, more consistent batch-to-batch performance, and no supply chain dependency on a wild animal population subject to ecological decline. In May 2025, the US Pharmacopoeia officially adopted a chapter placing RFC on equal regulatory footing with LAL for endotoxin testing. The technology exists, the regulatory barriers have been cleared, the science is settled. The only remaining obstacle is institutional inertia, an industry that has used horseshoe crab blood for half a century and moves slowly even when every measurable indicator says the alternative is at least as good and often better. The horseshoe crab is not a charismatic animal. It does not have the majesty of a whale or the menace of a great white shark. It looks like an artifact from a forgotten military project, a low, armored dome with a spike on the back, trundling across the mudflat on 10 stubby legs, eating whatever it finds in the sediment, sensing light from 10 different eyes, bleeding blue when cut, and spawning in numbers that blacken entire beaches every spring. It does not inspire the awe that a Tyrannosaurus does, or the dread that a megalodon does, or the fascination that a Dunkleosteus does. But it has outlived all of them, every single one. It has outlived every dinosaur, every pterosaur, every marine reptile, every giant shark, every armored fish, every trilobite, every ammonite, and every other organism that was once considered the dominant life form on Earth. It was alive when the first coral reefs formed, and it was alive when they collapsed. It was there when the Devonian forests spread across the continents and changed the atmosphere. It watched the Permian world burn and the Triassic world rebuild. It was there when the first dinosaur took its first step, and it was there 66 million years later when the last one took its last. It watched the mammals inherit a shattered planet, watched one lineage of primates develop tools and language and civilization, watched that civilization spread across every continent and begin harvesting its blood in clean white laboratories to keep its pharmaceuticals safe. And now that one species, the newcomer, the 300,000-year-old latecomer, to a 4.5 billion-year-old planet, is the first thing in 450 million years that the horseshoe crab might not survive. Not an ice age, not a supervolcano, not an asteroid. Us, a species that has existed for an eye blink of geological time, threatening to end a survival record that spans nearly half the history of complex life on Earth, not out of malice, but out of inertia, because switching to a synthetic alternative to its blood is an inconvenience that a trillion-dollar industry has been slow to prioritize. 5 mass extinctions could not kill it. The question now is whether we will.